ومعروف عني يعني في شيء سر معروف عني بمحاضرات البايوكيمستري خاصة بالموديولز شوي متعاطف مع الطلاب، كونه التايم ما بسمح انه نخش في تفاصيل كثير كبيره، مطلوب منا نغطي تشابتر في كل موضوع. فهذا الوقت الحقيقه 50 دقيقه او ساعه ما بكفي نغطي تشابتر، فبنحاول نركز على اهم الاشياء الخاصه بالبايوكيمستري ثينجز. هلا بدي ابلش، انا اعتقد انه هو بسجل لحاله اوتوماتيك. خليني اعمل شير للسكرين. اوكي that's it. And here we go. ان شاء الله تكون الشاشه ظهرت عندكم الباوربوينتس.
ظهرت دكتور.
طيب تمام، ثانك يو. طيب هلا وعد انه ما يكون في اسئله من السنوات السابقه او مكرره، فلذلك انتبهوا معي، يعني يفضل حضوركم رغم ان المحاضره مسجله، يعني راح تسمعها باي وقت اخر، ولكن الانتراكتيف sometimes is a good memory for recalling. وخذ عندك كومنتس على النوت بوك الموجود عندك، تنسى الاشياء راح نحكيها. هلا هذا الشابتر او هذا التوبيك، احنا في عندنا 2 lectures in the CNS1 regarding the biochemistry issues. هلا المحاضره الاولى بتحكي عن الميتابوليزم of neurotransmitters. اكيد انتم بتغطوهم في اماكن اخرى في الباثولوجي، sometimes in the pathology and the physiology most of the time, and they are well described over there when you talk about the action of these neurotransmitters. What we are going to talk about in the biochemistry part is the metabolism of these neurotransmitters and how they are synthesized in general. So first of all, let's discuss or have an overview of the outline of the things that we are going to discuss today. First of all, we will discuss the synthesis and degradation, which means the metabolism of what's known as gamma aminobutyric acid or GABA molecule. Then the synthesis and degradation of dopamine, epinephrine, and norepinephrine, as well as the metabolism of serotonin. Then glutamate metabolism as an amino acid, which is because it's important for the generation of the GABA molecule. And then we will have kind of short overview and description of the brain peptides as neurotransmitters. We will just have an overview of these molecules in order to have an idea of these molecules, how they are synthesized. In order to understand the difference between such kind of modified amino acid and other amino acids, we have to discuss the basic structure of the amino acids that we already knew. Basically, the amino acid consists of, let me change the line, the color of the line. Okay. So if you remember, the amino acid structure consists of, why it's black? So the structure of the amino acids consists of the alpha carbon, which is the central carbon here, and we have the carboxyl group and the amino group in the left side, as well as the hydrogen, and we have our group on the other side of the amino acid. So these amino acids, or the amino acids that we discussed before, most of them are alpha amino acids because we have alpha carbon that is conjugated with the carboxyl group and amino group, and this carboxyl group is known as alpha carboxyl group, and the amino group is called alpha amino group. هذا التصنيف عشان نعرف انه الكاربوكسيل جروب والامينو جروب مرتبطين بالالفا كربون. هلا الالفا كربون معناها انه في عندنا بيتا جاما and so on. So the other carbons that will be present in the side chain or the R group will be nomenclatured as alpha, beta, gamma, and so on. So if you have any modification on these groups, the modification will belong to the carbon according to its alphabetical order. For example, in the non-alpha amino acids, in these examples, we have, for example, beta-alanine, which means that the amino group is conjugated with the beta carbon because this is the alpha carbon, which is represented here. And here is the carboxyl group موجود هون. And you don't have this branch of the amino group. You have here alpha, beta, and then amino group. It means that we are going in this direction. So that's why we have to understand where it comes from as gamma amino acid butyric or gamma amino butyric acid name. Because we have these nomenclature issues that are related to the alphabetical order of the carbons in the amino acids. As well as this example here, we have what's called beta amino isobutyric acid. This is the isobutyric acid, and because we don't have alpha amino group that is conjugated with the alpha carbon, we have here amino group that is conjugated with the beta carbon, and the alpha carbon is busy with the methyl group in order to generate a new molecule, completely new molecule, which is known as beta amino isobutyric acid. Now going to our molecule, which is known as gamma amino butyric acid or butyrate or GABA. If you count the carbons, this is carbon alpha, beta, gamma, and the amino group is conjugated to the gamma carbon, which is carbon number 8. Okay, and this is the structure again in details. This is the alpha carbon, which was actually the alpha carbon, and here we have beta, gamma, and the amino group is conjugated with the gamma carbon, and that's why it's called gamma amino butyric acid or butyrate or GABA molecule. In fact, you know actually that this kind of molecule is brain inhibitory neurotransmitter that alter transmembrane potential difference, and the molecule itself is generated by a reaction known as decarboxylation reaction of the glutamate molecule. So what you have to do, or what the body does actually, is decarboxylation reaction, which means that you are removing the carboxyl group from the side chain in order to generate such kind of molecule. So moving forward to see what's going to happen in order to generate our molecule or our neurotransmitter here. This is the glutamic acid or the glutamate molecule. This is the carboxyl group, and this is the side chain of the glutamic acid, and this is the amino group. This is the alpha carbon, and we have here the carboxyl group of the alpha carboxyl group actually, and this is the alpha amino group. When you do decarboxylation reaction, you will generate such kind of molecule. Actually, you are removing the carboxyl group, which was here, in order to generate such kind of molecule, and it will be known as gamma aminobutyric acid. So this reaction, as I mentioned before, is catalyzed by glutamate decarboxylase, and decarboxylase means that you are going to generate carbon dioxide as a free carbon because it's decarboxylation reaction. And most of the decarboxylation reactions require the presence of such kind of coenzyme. So B6 or vitamin B6 is the coenzyme that is required for the most of the decarboxylases enzymes that we have in our body or in our metabolic pathways in our body. Basically, we know that the glutamate molecule is very important amino acid as a residue because it can be generated from many sources like the Krebs cycle or TCA cycle, as well as deamination of the glutamine, okay, in order to synthesize the glutamate. So this kind of reaction is deamination reaction. You are removing the amino group in order to generate glutamate, as well as transamination reaction. And you can generate glutamine as well from the glutamate by glutamine synthase or synthetase enzyme because it needs ATP in order to generate such molecule. Now, our molecule, which is the glutamate, is a precursor for the GABA molecule as a neurotransmitter. And as we discussed before, it requires the presence of such kind of coenzyme because it's decarboxylation reaction. And then the GABA molecule can be metabolized, as we will discuss in the next few slides, to generate what's called succinate semialdehyde molecule by transamination reaction, and it's catalyzed by 4-aminobutyrate transaminase, because you are going to transfer amino group from one molecule to another in order to generate succinate semialdehyde molecule, and it will be a precursor for one of the molecules that will be integrated in the Krebs cycle or TCA cycle, and this reaction is catalyzed by succinate semialdehyde dehydrogenase reaction or enzyme. So, discussing more details about the glutamate metabolism as well as generation of the gamma aminobutyrate molecule. As you can see here, we have the reaction that we described before, the conversion of glutamate to gamma aminobutyrate molecule by pyridoxal phosphate, which is representing the B6 or vitamin B6. Okay, in order to have decarboxylation reaction and removal of the carbon from the molecule in order to generate our molecule. Okay, so the enzyme again is known as decarboxylase or glutamate decarboxylase enzyme. The other side of the reaction can be the generation of what's called alpha ketoglutarate from glutamate and conversion of one of the keto acids to another amino acid. هلا هذا الرياكشن اللي هون مشهور باسم transamination reaction. Transamination reaction واحد من الرياكشنز اللي يمكن ناقشناهم، اكيد ناقشناهم، ولكن بحكي كلمتين يمكن عشان اللي مش متذكر يتذكر انه the transamination reaction in general they are reversible reactions that involve conversion of one amino acid اللي هو الجلوتاميت to keto acid، والكيتو acid اللي generated من الالفا من الجلوتاميت is alpha ketoglutarate. This is the keto acid of the glutamate, and the conversion of another keto acid to an amino acid. So what you have, or what you are going to do, is conversion of a keto acid to an amino acid and conversion of one amino acid like glutamate to keto acid, or vice versa, keto acid to an amino acid and amino acid to keto acid. So this kind of reaction is catalyzed by transaminase enzyme, or transaminase is general description of many enzymes. في عندنا مجموعة من الانزيمز حسب نوع الامينو اسيد اللي involved في هذا الرياكشن. هلا في الواقع انه هذا الرياكشن هو بحد ذاته الجلوتاميت للالفا كيتوجلوتريت او العكس اللي هو reversible reaction is the paired reaction with the other transamination process of other amino acids. يعني ممكن انت تحضر اكثر من امينو اسيد بهذا الرياكشن باستخدام نفس الرياكشن الموجود هون. هلا بالضروره يكون في عندي pyridoxal phosphate as a coenzyme that is required for such kind of transamination reaction. خليني امسح بس الاشياء اللي هون. تمام. هلا تعرفنا على شغلتين او two reactions that are involving the glutamate metabolism، واحنا بالنسبه لنا مهم جدا اللي هو موضوع generation of GABA molecule, and the GABA molecule has to be catabolized again because it's present there, it's inhibitory neurotransmitter, so it has to be catabolized, and the catabolism involves the conversion of alpha aminobutyric acid or butyrate to succinate semialdehyde molecule as we discussed earlier, and this reaction requires the presence of the same coenzyme اللي أشرنا له قبل شوي, and it's deamination reaction. In fact, the pyridoxal phosphate is involved in such kind of reaction. هلا في بعض الأحيان في عندنا kind of rare genetic disorders that are related to the GABA metabolism or actually inhibition of the process of the GABA degradation molecule, which will result in the accumulation of such kind of molecule and causing certain neurological disorders. And this is fortunately a rare genetic disorder that is related to the deficiency in the aminotransferase or GABA aminotransferase enzyme. Such kind of reaction. The last reaction here of the metabolism is the conversion of alpha ketoglutarate to succinate semialdehyde molecule, which can be converted into two molecules. One of them is the succinate, which is very famous molecule in the Krebs cycle. So we can feed the Krebs cycle by such kind of metabolic pathways. It means that you can generate energy from glutamate in this kind of metabolism, اللي هو catabolism of the alpha butyrate or catabolism of amino butyrate molecule. On the other hand, you can generate what's called hydroxybutyrate by lactate dehydrogenase enzyme. It is similar to the lactate dehydrogenase that we already knew, اللي هو بيتعامل مع اللاكتيت والبيروفيت molecule, conversion of lactate to pyruvate and vice versa. This reaction is one direction reaction which will generate hydroxybutyrate molecule which can be used for another metabolic reactions in the body. And because it's dehydrogenase, it means that there is an involvement of reducing agent, and here we have also oxidizing agent on the other hand. هلا هذا reducing عملية reduction هون عملية oxidation because we have an AD that is involving the conversion of succinate semialdehyde molecule to succinate. If you inhibit this enzyme, there will be accumulation of what's called 4-hydroxybutyric acid molecule, and it will appear in the urine and in the CSF fluid, and it will cause certain neurological disorders because of the accumulation of such kind of molecule, because of the deficiency in the metabolism of the let me say intermediate product of the glutamate or GABA molecule metabolism. So these are the most important things that are related to the metabolism of the GABA molecule or gamma aminobutyrate molecule, and just to understand the precursor and the output product of the catabolized molecule. So I hope it's clear. Moving to the next neurotransmitter that can be generated from amino acids. And that's why we are studying the biochemistry of these molecules, because they are involving the metabolism of amino acids. And this is called special products of amino acids or nitrogen component other than the proteins that can be used to generate such important molecules. For example, here we have 4 molecules that can be generated from the tyrosine, which is one of our amino acids that are coded for protein synthesis. And eventually it will generate what's called epinephrine molecule. And the generation of epinephrine involves 4 major steps. The first one is called hydroxylation step, and then decarboxylation. So in the first step, you are adding hydroxyl group, and then you are removing the carbon dioxide or the carbon by decarboxylation process. And then we have oxidation process. And finally, we have process of methylation or addition of methyl group. Okay, so let's discuss them in an easy way. The first step is conversion of tyrosine to Dopa molecule, and the new group is the hydroxyl group, and it's generated by the action of tyrosine hydroxylase. And this reaction requires the presence of this coenzyme, which is known as tetrahydrobiopterin. يسموه بتحول إلى dihydrobiopterin as a coenzyme. The next step is the decarboxylation by conversion of Dopa to dopamine. And the difference is clear and obvious. You have amino group instead of the carboxyl group. So you have decarboxylation and leaving the amino group اللي كانت هون صارت هي على الطرف بدل carboxyl group. So it will be here. So conversion of Dopa to dopamine by decarboxylation requires the action of Dopa decarboxylase enzyme. وزي ما شفنا قبل شوي بالجابا molecule metabolism, the decarboxylation relies on the presence of pyridoxal phosphate coenzyme, which is known as vitamin B6 as well. Okay, then conversion of dopamine to norepinephrine by the action of dopamine oxidase enzyme. And it's very important to remember the presence of these coenzymes that are required for the oxidation process, which is the copper as well as vitamin C or ascorbic acid. And the final step, which can be conducted in the adrenal medulla by the conversion of norepinephrine to epinephrine. And the difference is the methylation process, deamination, methylation. It means that also actually methylation of the amino group. There is no deamination. You are adding the methyl group to the molecule. So the methylation process is catalyzed by phenyl ethanol amine methyltransferase enzyme. So in order to add this methyl group, you have to have a source of this carbon, which will be S-adenosyl methionine, which will be converted to S-adenosyl homocysteine molecule. So this molecule is known as carbon carrier that will be used in order to generate or to transfer the methyl group from it to another precursor, which is in this case the norepinephrine, in order to generate the epinephrine molecule. So you have to be familiar with these steps that will convert tyrosine to the eventual molecule, which is the epinephrine molecule. Okay. So basically what we learned is that, the neural cells can convert tyrosine to epinephrine and norepinephrine. Dopa is also intermediate in the formation of melanin in the melanocyte. It's another metabolic pathway that will be used in order to generate melanin stain in the melanocytes. The other molecule that you have to be familiar with, and it will be generated from another amino acid, and this amino acid is known as tryptophan molecule. And the hydroxylase enzyme is responsible for the generation of what's called 5-hydroxytryptophan and finally 5-hydroxytryptamine molecule. So this is our target or our final product to be synthesized, which is known as serotonin, and it's potent vasoconstrictor molecule as well as stimulator of smooth muscle contraction. So this molecule is very crucial molecule for many actions, and one of them is the feeling of happiness, for example, because it's vasoconstrictor molecule as well as stimulator of the smooth muscle contraction, and it's generated from a precursor molecule, which is the tryptophan amino acid. So you have to be familiar with the metabolic pathway. هون في عندنا under decarboxylation reaction you have to be familiar with as well, and to know that the generation of serotonin also requires decarboxylation reaction of the intermediate amino acid, which is the hydroxybutyrate or hydroxytryptophan molecule. Now, the serotonin itself, if it stay in the body, you will feel high all of the time. So it has to be catabolized. So the serotonin can be used in order to generate the melatonin, for example, molecule. And I hope that you are familiar with the function of the melatonin. حدا يقول لي شو الفانكشن تبع الميلاتونين؟
بالسليب.
Yes. Thank
You
you.
that sleeps like it.
Thank you. So if you go to walk, for example, you will generate serotonin, which make you feel happy. And then you can go to sleep if you have such kind or some types of depression or feeling of, let's say, any emotional stress, for example. So walking is very helpful, or the process of the generation of the serotonin is very helpful in order to feel happy as well as to go to sleep. So this is the secret of the action of the serotonin as melatonin. Now, for the individuals who are suffering from depression, for example, they use such kind of iproniazid molecule or iproniazid, as you wish, pronunciation, because it works on the enzyme or complex that is known as monoamine oxidase complex that will catabolize the serotonin. So it will cause some kind of accumulation of serotonin, and that makes these individuals happier, or actually it works as antidepressant agents. On the other hand, if you don't have such kind of inhibition, the normal pathway will go through the excretion of the serotonin as conjugated molecule. I'm not so interested, or it's not that important to remember these names of these molecules, just to know that the serotonin can be excreted from the body by conjugation process that requires different types of kind of deamination, for example, or addition of methyl group. So just to know that we can excrete such kind of molecule from the body. And as I described before, we can generate melatonin from the same molecule, and another kind of conjugation can be used in order to excrete or to get rid of the serotonin. Otherwise, it will be kind of crazy emotions when you have such high concentration of serotonin. يعني ممكن تكون قاعد في قاعده جديه وتبلش تضحك وانت مبسوط والناس معزه، بتسبب مشاكل. فمش بالضروره دائما يكون نفس المولكيول موجود، ومش بالضروره دائما يكون يعني اوفر هابي خليني اسميها. So we have to degrade the excess of these molecules. هلا واحد الاشياء الغريبه جدا ممكن يستخدموها في الدياجنوسيس. They notice that, and I like this example, اللي هو special secretion of serotonin from kind of carcinoids, which is known as argentophenoma, for example. It's a kind of tumor, and I guess it's kind of colorectal tumor that is responsible for the production of serotonin in high concentration, meaning overexpression or overproduction of the serotonin of such kind of tumors. On the other hand, the melatonin can be taken by many tissues. One of them is the brain, followed by hydroxylation process that will cause conjugation of such kind of molecule in order to be depleted from the body or secreted from the body. Let's talk about some biochemical issues that may be annoying for some of you, but we have to talk about it in brief. And in fact, okay. We discussed this before many times. The whole Krebs cycle itself by its components. And we mentioned at a time previously, some amino acids can be used to generate energy. And this is how these amino acids will be used to generate energy. It's not kind of miracle. It's, it's not that difficult to understand the metabolism of the amino acids in order to generate energy. Keep in mind, we don't rely on amino acids to produce high amount of energy. We could, as I mentioned many times earlier, إنه ممكن إحنا نصنع أو ننتج طاقة من الميتابوليزم أو الكاتابوليزم of the amino acids، بس إحنا ما بنعتمد عليهم كمصدر أساسي للطاقة زي الكربوهيدرات وزي الليبيدز. وهذا السر اللي بيخلي الليبيدز والكربوهيدرات إنه stored in the body in order to be fuel of the body for energy production. بينما الامينو اسيدز احنا ما بنخزنهم، احنا بنعمل منهم structures، نعمل منهم metabolites for example، والسبب انه they are not good for energy production. طيب شو التناقض هذا؟ يعني انت بتحكي انه not good for energy production and we can use them to produce energy. هلا المعنيين لا يتناقضوا مع بعض. احنا بنقدر نستخدمهم في حالات وظروف معينه من ضمنها الستارفيشن او الفاستنج، تمام؟ the other things is the metabolism of these amino acids اللي بيكونوا موجودين نتيجه turn over in the body. turn over معناها في عندك بروتينات بتصير لها degradation، بروتينات بتصير لها production، وبالتالي هذا turn over بتطلب انه البروتينات بتصير لها degradation تفرز احماض امينيه، وهذول الاحماض الامينيه راح يكون لهم destination معين. اذا ما استخدمتهم في التصنيع مضطر ان استخدمهم في شيء اخر او اعملهم excretion from the body. So we can use them، ولكن ما نعتمد عليهم. السبب بسيط جدا حكيناه يمكن سابقا، اللي هو انتاجهم للنيتروجين. اذا بتعمل degradation للامينو acids they will produce high amount of nitrogen. The nitrogen will be produced as an ammonia molecule which will be toxic for the body. So that's why they are not preferred to produce energy, or they are not the first choice to produce energy. However, we can use the ammonia or metabolite the ammonia to produce urea, for example, in order to reduce the toxicity of the ammonia. But it's not the choice to produce energy and then making another pathway in order to get rid of the ammonia. So the ammonia or the ammonia metabolism to produce urea is protecting mechanism to get rid of the toxic ammonia that we have from the metabolic pathways, not to produce energy. So going back to the story that we started with, اللي هو الامينو اسيدز احنا بنستخدمهم لانتاج الطاقه. هذول لانتاج for example sometimes energy and glucose, they can be called ketogenic amino acids or glucogenic amino acids. هلا الكيتوجينيك امينو اسيدز هم الامينو اسيدز اللي على سبيل المثال بعطوني كيتون مولكيول زي الاسيتيل كوي مولكيول، هذول بنسميهم كوتوجينيك امينو اسيدز. والامينو اسيدز اللي بيعطوني مركبات ممكن نستخدمهم لصناعه الجلوكوز بنسميهم جلوكوجينيك امينو اسيدز. هلا المهم في هذا الموضوع كله انه we have kind of integration between metabolic pathways, metabolism of amino acids as well as metabolism of glucose by glycolysis and Krebs cycle. The most important thing here to know that we have integration between the metabolism of glutamate, which can be generated from these amino acids, arginine, histidine, glutamine, and proline, in order to produce alpha-ketoglutarate by transamination reaction. Transamination معناها انه انا بجيب هون كيتو اسيد وبحوله لامينو اسيد. خليني ارسمها مشان اتذكر هذا الموضوع. It's very important. So we have transamination reaction. Okay. So the transamination reaction means that you are going to have what's called alpha keto acid, and you will generate another amino acid. And here we have transamination reaction or transaminase enzyme. So you will generate alpha-ketoglutarate, which can be metabolized in the Krebs cycle itself in order to produce energy. So this is the integration between the glutamate and the Krebs cycle metabolic pathway. And the other thing here, just to understand how we generate the glutamate from these amino acids. وهون أنا بديش أصعبها عليكم كثير. I'll try to be nice with you in order to just understand the general concept. So basically, this is the reaction that we described before, اللي هو الجلوتاميت يتحول لألفا كيتوجلوتاريت by transamination reaction. Transamination معناها بالtransaminase enzyme. And it is, in this example, they took the conversion of pyruvate to alanine. On the other hand, you can generate pyruvate from alanine and convert alpha ketoglutarate to glutamate. And the glutamate can be used to synthesize the glutamine by amination reaction. Amination means that you are adding amino group. Now, the glutamate itself can be generated from the glutamine molecule by what's called deamination reaction. And this is catalyzed by glutaminase enzyme. And the glutamine is the carrier of the amino group that will be generated in other tissues or in the body itself. Now, as I described before, we have these 4 amino acids that will be converted to glutamate and then alpha-ketoglutarate. Let's discuss them generally, how we convert, for example, proline to glutamate and then to alpha-ketoglutarate. هذا reaction صار مفهوم بالنسبة لنا اللي هو transamination reaction. And I'm not so interested to make you knowing these details of the conversion of proline to glutamate, just to know that we have these kind of complex reactions and they require many dehydrogenases in order to convert proline to glutamate. So it's very tough oxidation process in order to generate glutamate from the proline. While here we have another kind of metabolic pathway that will convert arginine, which is non-amino acid, by arginase, وهذا بذكرنا بالurea cycle, which will generate ornithine. And the ornithine is another metabolite as an amino acid. هو بالنسبة لنا amino acid ولكن مش من amino acids الموجودين في البروتينز. We are very familiar with this molecule. And the ornithine will be converted to what's called glutamate gamma cymaldehyde molecule. At the same time, you can convert alpha ketoglutarate to glutamate. So this is our molecule here, okay, by ornithine delta aminotransferase enzyme. Okay, so the arginine can be converted to glutamate molecule by such kind of reaction. Here we have another source for the generation of glutamate, which is the histidine, and we have many steps that include histidinase or histidase enzyme by removing the amino group, or deamination reaction, and urokinase enzyme, and then finally we have the generation of glutamate molecule. Again, I'm not interested in these much details about the conversion of the histidine to glutamate because our course is not metabolism of amino acids. We are talking about the metabolism of the neurotransmitters. In order to make the story easier, we have to understand the following. The proteins are the source of the amino acids that we have in our body, as well as degradation of tissues and the they will make what's called amino acid pools that can be used to generate many different molecules that could involve the production of energy or production of ketone bodies, for example, nitrogen containing compounds, or the glutamate molecule that we are interested in, or they can be glucogenic amino acids. So these are the things that can be used from, or that can be fate, let me say, for the amino acids that we have in our body. Any defect in these metabolic pathways will cause accumulation of these amino acids and will be associated with another disorder. وهذول مرتبطين بشيء اسمه الامينو acid metabolic disorders. Now moving to something slightly different, اللي هو generation of other types of neurotransmitters, and they are known as brain peptides. And it's obvious that they consist of peptides, small chain of amino acids, and they are numerous. They work as neurotransmitters in the brain, and they classify to 3 types, like opioids, substance P, or stereopeptide hormones. All of these molecules, or all of these neurotransmitters, are generated from peptides. So they are proteins that will be, or large peptides that will be cleaved in order to generate such molecules as brain neurotransmitters. And the opioids classified or includes, in fact, 3 types of opioids or opioid peptides, اللي هو endorphins, enkephalins, and dynorphins. Okay, so in order to understand the relationship between these molecules, we have to know that these molecules, because they are peptides, they have to have genes that will generate them. So basically, we start with production of pre-pro opio melanocortin, for example, or pre-pro enkephalin A, or pre-pro dynorphin. It means that you are making big peptide, and then you cleave these peptides in order to generate these opioid molecules. And we will talk about the other molecules one by one in the next few slides. Let's start with the endorphin, which is the first opioid molecule. And the endorphin is generated from the pre-pro peptide molecule that will be cleaved by removing the signal peptide in order to generate what's called POMC, اللي هو اختصار ل pro opio melanocortin molecule. And then the cleavage of this large peptide will generate ACTH, which is the very well-known hormone, as well as the beta lipotropin. And then finally, we will generate endorphin and lipotropin hormones from, or by cleaving the last molecule that can be cleaved by cleavage enzymes. So this is the basic structure of the human beta endorphin molecule that will be generated from the large pre-POMC, if you would, POMC molecule by cleaving this large molecule. Then the enkephalins generated from, enkephalins means that you have much more than one enkephalin molecule that will be generated in the same way by cleaving a big peptide after removing of the structure of the signal peptide. بتلاحظ هون لما بكون في عندي signal peptide we have prefix of pre. كلمة pre معناها إنه لا زالت signal peptide اللي بيوم من الأيام صنعها البروتين أو كونها البروتين حتى تدله وين يعمل sorting لهذا البرودكت، ففائدتها فقط signaling اللي هو دلالة وتعمل tracking للبروتين or direction of the protein in order to be sorted in a certain way. Then you will have these final products of active components of enkephalins, and in the physiology part, you discuss the function of these enkephalins. Dynorphin or dynorphins, actually, because we have more than one type, also consist of a short peptide molecule. Just to know the structure of the endorphin in general, I will not ask about the details of these molecules. Moving to the second type of the brain peptides, it was substance B or P, sorry, which consists of such kind of looks like as a complicated structure، بس لأنه إحنا راسمين الديتيلز ده الأحماض الأمينية الموجودين هون، مكون من 2 4 6 8 10 11 بيبتايد، وهذا مذكور اصلا في السلايد الاول هون. It consists of 11 amino acids or residues. Okay. And this kind of molecule is related to the inflammatory and pain feeling. Just to know that it consists of such kind of short peptide, vasopressin and oxytocin, very similar hormones or peptide hormones that are generated by the pituitary gland, and they have the similar structure, not identical structure, because we have some differences here at position number 3 as well as position number 8. Okay, so just to know the difference, as well as the presence of the SS bond in the same position between residue number 1 and residue number 6. This is very important in order to understand how it works or how it be synthesized. So here we are going to finish our lecture today, which I think it's very light lecture. It's like a dissertation, much more than being a heavy lecture. So if you have any questions, the floor is yours.
دكتور.
Yes.
بدي أسأل سؤال. لما حكينا عن الجلوتاميت ميتابوليزم، إذا في إمكانية نرجع للسلايدز بس.
تمام. Yeah, this one.
لا، مو هاي اللي بعدها.
لا حتى يصير نحن نروح بعديها. هلا آخر سلايد بالميتابوليزم الجلوتاميت هون، وفي سلايد هون.
دكتور السلايدز لقدام مش هون.
تحكي عن السينثيسيس. Yeah. Okay. Wait. Yes.
دكتور هون لما حكينا آخر خطوة الجلوتاميت لما يتحول لألفا كيتو جلوتاريت، هلا
yes.
هون مكتوب إن ترانز أمينيز بيحول البيروفيت لألينين.
لا ألينين exactly.
طب ما هو البيروفيت أصلا مش أمينو أسيد، فكيف بدي أحول؟
مش أمينو أسيد. كويس السؤال. هلا مين الأمينو أسيدز هون بالرياكشنز؟ الجلوتاميت right؟
تمام.
So you are converting amino acid to alpha keto acid right؟ وهون بتحولي العكس alpha keto acid، هذا keto acid بالمناسبة، to amino acid، لأنه مستحيل يكون على نفس الرياكشن to amino acids because you are transferring amino group from glutamate تعطيها للبيروفيت، تتحول البيروفيت لألانين، ولما فقد الجلوتاميت الامينو جروب تحول لألفا كيتو جلوتاريت. فالترانسامينيشن رياكشن always convert amino acid to alpha keto acid. كلير؟ وهذول الرياكشنز تبعون او الانزيمز خلينا نحكي اللي هم مسؤولين عن الترانسامينيشن، اعتقد ناقشناهم في مكان اخر لما حكينا عن الالانين امين ترانسفيريز انزيم اللي هو ALT في حالتنا هاي. تمام؟ وفي عندنا الاوكزالو استيت ترانسامينيز انزيم اللي هو بحول الاوكزالو استيت لاسبارتيت، فعشان هيك بصير اسم الانزيم هناك AST. في عندنا تو انزيمز AST و ALT، هذول مؤشرات للليفر فانكشن. When we talked about liver function test, I think in the GIT module. Any other question? Okay. هلا في سؤال راح يتكرر كثير اللي هو موضوع انه هذا بالميد وهذا بالفاينل. احنا بنحكي عن محاضرتين يا جماعه، وبالتالي مش منطقي انه يعني محاضره تمتحنها بالميد ومحاضره بالفاينل. الاسئله راح تيجي على الميد من المحاضرتين وعلى الفاينل من المحاضرتين. يعني مش بحاجه لكل هالتعقيد الا اذا شاء الله امرا اخر يعني، لانه CNS اعتقد هو واحد من الشورت كورسز. طيب نلتقي حسب الاوتلاين الموجود معي ان احنا راح نلتقي ب3 الشهر 3/3 يوم الاربعاء القادم ان شاء الله. Good luck for all. Thank you. هلا بدي حدا بس ابعث له، انا مش عارف مين الطالب الCR اللي بالنسبه لكم اللي دفعتكم، ابعث له الريكورد والسلايدز. السلايد انا معكم سابقا بس بضل الريكورد. راح ابعثه للدكتوره ليلى وابعثه للCR. يعطيكم الف عافيه.